Rise and shine, edukalidad. Great morning, students and dear listeners. Welcome to the new normal of our class on air here in DXVD 101.3 FM. This is Teacher Shermay from the Pitan City National High School for grade 9 science lesson for the week 2 of October. Grade 9 students, are you ready? In this session, let's study non-Mendelian pattern of inheritance or the non-Mendelian genetics. But first, let's revisit Gregor-Johann Mendel genetics, which we call Mendelian genetics or the non-Mendelian pattern of inheritance. Mendel's model of inheritance assumes that each trait is controlled by a single gene. Each gene has only two alleles or two versions of gene, which can be either dominant or recessive, and dominant allele is always expressed over the recessive allele. In real life, most genes or traits do not meet or follow these criteria, and this is where the non-Mendelian genetics or the non-Mendelian pattern of inheritance comes in. Now, what does non-Mendelian mean? What is meant by non-Mendelian? When we say non-Mendelian, traits do not follow or obey the rule of simple dominance and recessiveness. Under the non-Mendelian traits, some traits have more than two alleles or variations. Some traits are the result of many genes that interact together, and some traits are passed on via the sex chromosomes. When we say sex chromosomes, in humans, these are the 23rd pair of chromosomes that determine the gender or the sex of the individual. For the males, sex chromosomes, it is XY in uppercase or capital letter. For the female sex chromosomes, it is XX in uppercase or capital letter. Some examples of non-Mendelian inheritance pattern are number 1, incomplete dominance, number 2, codominance, number 3, multiple allelism, number 4, sex-linked traits, number 5, sex-influenced traits, and the last, number 6, the sex-limited traits. Actually, there are more examples of non-Mendelian patterns, but we will just focus on these 6 patterns in our study. So, what is incomplete dominance? When we say incomplete dominance, it is where two dominant and different alleles of the parents blend or mix to form a hybridized intermediate phenotype. Biologists call this phenomenon blending phenotype because it is sort like mixing of two paint colors resulting in a blend or a mix. Classic example that shows incomplete dominance pattern of inheritance is the flowers of 4 o'clock and snapdragon. When the dominant red petal is crossed with the dominant white color of the 4 o'clock, the result is a pink petal color of the 4 o'clock flower. And the same is true with the snapdragon flower. The pink petal color of the 4 o'clock flower is hybrid or heterozygous because it carries the allele for the red petal and the allele for the white petal. Again, when we say heterozygous, it means it has two different alleles or two contrasting alleles which are dominant. Remember, in the context of incomplete dominance, again, when we say heterozygous, it means it has two dominant contrasting alleles or alleles which are different and dominant. So alleles mean versions or variations of a gene. In setting up incomplete dominance problem, we use all capital letters for allele notation or genotype, since the parents' alleles are all dominant here, and we use two different letters, one per variation. Example, in snapdragon petal color, we use RR in uppercase or capital letter for the red phenotype. We use WW in uppercase or capital letter for the white phenotype, and RW in uppercase or capital letter for the pink phenotype, which is heterozygous. Remember, we have a pair of alleles which we call genotype for every phenotype or color trait. One allele from the male parent and the other allele from the female parent. When a tan color is crossed with black color, the result is tabby. When we say tabby, it means there is black and tan color together in the phenotype of the offspring. So here, there is no mixing, no blending, no gray, or no intermediate effect of the phenotype. What you see in the phenotype of the offspring is the individual color of the two dominant traits of the parents that are equally expressed. You can see the black and tan color together in the phenotype of the offspring. More examples of the codominance include roan coloration in cows. When the red coat cow is crossed with the white coat cow, the offspring phenotype will have the red and white coat together. The expression of AB blood type in humans is another example of codominance pattern of inheritance. The alleles or genotype of a trait in codominance are always expressed in capital letter or uppercase letter, just like the alleles in incomplete dominance, since all the alleles here are dominant. For cow's coat color, both the red and the white are dominant. To express the genotype of the cow's coat color, the red coat phenotype is expressed in RR in uppercase or capital letter. The white coat cow phenotype is expressed in WW in uppercase or capital letter, and the roan cow phenotype is expressed in RW in uppercase or capital letter. So note, the roan coloration is a heterozygous genotype because of the presence of the two different alleles in the phenotype of the offspring. Now let's proceed to multiple allelism. What is multiple allelism? Multiple allelism exists when three or more possible alleles or versions of genes are present for a given trait. An example of this in humans is the ABO blood type. ABO blood type is controlled by the three alleles, which are the A, B, and O allele. There are rules for blood types. Number 1, A and B alleles are codominant, meaning they both show in the phenotype of the offspring. So when the A allele is paired with another A, the result is type A homozygous. When the B allele is paired with another B allele, the result is type B homozygous. And when the A allele is paired with the B allele, the result is type AB heterozygous. So again, when we say homozygous, it means the genotype has two identical or the same alleles. And when we say heterozygous, it means the genotype has two different or contrasting alleles. Number 2, the A and B are dominant over O, meaning when the O allele is combined with the A or the B allele, it is not physically expressed because the O allele is recessive. In writing blood alleles, for simplicity, we can use the letters A, B, and O. Or we can use the other version of the blood allele notation. For example, for the A allele, we can use capital letter I with superscript A in uppercase. And for the B allele, we can use capital letter I with superscript B in uppercase. And for the O allele, we can express it using small letter I or the I in lowercase. Take note, the capital letter I here stands for the word isoagglutinogen, which is another term for the antigen. Protein molecules found on the surface of the red blood cells and in the blood plasma, or the liquid portion of the blood, determine the blood type of an individual. There are two proteins in the blood. The first is antigens. Antigens are located on the surface of the red blood cells. The second is antibodies. These antibodies are located in the blood plasma or the liquid portion of the blood. When we say antigens, these are proteins in the blood which induce the production of antibodies. On the other hand, antibodies are the immunoglobulin protein in the blood which fight the antigens or foreign materials such as bacteria, viruses, and other harmful materials that enter in our body. Now, what are the characteristics of the different blood groups? The blood group A or the type A group have A antigens on the surface of the red blood cells and the B antibodies in the blood plasma. And this is the reason why the type A individuals can only receive type A and type O blood but cannot receive type B and type AB. The blood group B or the type B group have B antigens on the surface of the red blood cells and A antibodies in the blood plasma. And this is why the B group or the type B individuals can only receive type B and type O and cannot receive the type A and the type AB blood. The blood group AB have both A and B antigens on the surface of the red blood cells and no A or B antibodies in the blood plasma. And this is why the AB blood or the type AB blood is known as the universal receiver because it can receive all the different kinds of bloods from type A, type B, type AB, and type O. On the other hand, the blood group O or the type O have neither A or B antigens on the surface of the red blood cells but have both A and B antibodies in the blood plasma. That is why the type O blood is known as the universal donor because type O blood can be given to other blood types. Take note, type of blood donates to all blood types but receives only type O. Now let's try analyzing these problems. Number 1, how many possible genotypes for a person with a type A? The answer is there are 2 possible genotypes for a type A individual. The first genotype is AA and the second is AO. How about type B? What are the possible genotypes? So the same with type A, there are 2 possible genotypes for type B individual. And those genotypes are the BB for homozygous type B and the BO for the heterozygous type B. What 2 parents could produce the most diverse children as far as blood type is concerned? So who do you think are those parents that could produce variety of blood type in their offspring? Of course, the parents with heterozygous blood type, who are heterozygous type B and heterozygous type A. When these parents produce offspring, the offspring will have different blood types or variety of blood types from type A, type B, type AB, or type O. Now let's proceed to sex-linked traits. What are sex-linked traits? Sex-linked traits are traits found on the sex chromosomes of the individual. When the traits are found on the X chromosome, they are called the X-linked traits. And when the traits are found on the Y chromosome, they are called the Y-linked traits. So meaning, sex-linked traits can be X-linked or Y-linked. Remember, sex chromosomes determine the gender of the individual. For the male, it is XY, and for the female, it is XX. Examples of X-linked traits include disorders such as hemophilia, known as bleeding disease, and color blindness, in which the colorblind person cannot recognize green and red color. Both hemophilia and color blindness are recessive traits. So, for the females to have the disease of color blindness or hemophilia, the 2X chromosome of the female must have the copy of the recessive allele or must be defective with the allele in order for the trait to show up. If one X only of the female is affected or defective with this kind of sex-linked traits, the trait will not be expressed in the female. On the other hand, males have one X sex chromosome. Therefore, it only takes that one X to be affected to make males have an X-linked disease. And this is the reason why these sex-linked traits are more common in men than in women. Because for the men, it takes only one X to be defective. However, for the women, it takes for 2X to be defective by these recessive alleles. Now, is it possible for males to pass the linked traits to their sons? Can the father pass the traits, the color blindness and hemophilia, to his son? The answer is no. Males cannot pass the disease to their sons because there is no male-to-male transmission of the X chromosome. However, they can pass their defective X chromosome to their daughter. How about the mother? Can the mother pass the traits to her children? The answer is a big yes, because there is X chromosome transmission from the mother to son and from the mother to daughter. So in this case, the mother is the person responsible for transmitting the disorder to his son and not the father. Remember, the mother give X chromosome to both her son and daughter, and the father give only Y chromosome to his son and X chromosome to his daughter. Now let's go to the Y-linked traits. These Y-linked traits are genes found in the Y sex chromosome. They're also called Hollandric genes. Examples of this are the presence of excessive hair in the ears of some males and some forms of infertility. Y-linked disorders are transmitted directly from the father to son because there is Y transmission of the chromosome from the father to son, but this trait is not transmitted from father to daughter because there is no Y transmission from the father to his daughter. In solving sex-linked problems, we need to write the sex chromosome per gender of the individual. Second, figure out the genotype for the trait. For example, for color blindness, so the genotype is small letter C because it is recessive trait. And for the hemophilia, it is small letter H because it is a recessive trait. Then, we write the allele letter, which is the small letter C and the small letter H on the sex, which is the X chromosome. For example, for the trait color blindness, the normal female genotype is XX. For the normal female carrier of the gene color blindness, the genotype is X and another X with superscript small letter C. And for the colorblind female, it is X with superscript small letter C and another X with superscript letter C. For the normal male or the male with normal vision, the genotype is XY only. And for the colorblind male, the genotype is X with superscript small c and Y in uppercase. Remember that in sex-linked traits are either attached to the X or to the Y sex chromosome. Okay, let's proceed to the sex-influence trait. So what are these sex-influence traits? So there are four main features. Number one, they are located in the body chromosome or autosomes. Second, these traits are expressed in both sexes but in different variations or versions. Third, the expression of the trait is governed by hormone. And last, its dominance in heterozygous condition depends on the sex of the individual. Examples of these sex-influence traits are baldness trait and body hair. Both these traits are expressed in both sexes, the male and the female, but there is variation on how much of this trait is present in each sex. For example, baldness. So baldness is dominant trait for men and recessive trait for women. That is why this trait is common among the male population. In body hair trait, both males and females express the trait for body hair or facial hair, but obviously the expression appears different. For the male, the body hair is coarse, dark, and thick. For the female, the body hair is fine and thin. And what do you think is responsible for this difference in the expression of body hair? Of course, the effect of the hormones. Hormones dictate how much of this trait is being expressed in each sex or gender. For the female, we have the hormone estrogen, and for the male, we have the hormone testosterone. Now let's study the baldness pattern in human. The baldness trait is expressed by the allele B. For the female bald, the genotype is XX with homozygous dominant gene pair BB. When we say homozygous dominant, the alleles are written in capital letter or uppercase. For the female non-bald, there are two genotypes. The first is XX with heterozygous gene pair BB. When we say heterozygous, one of the allele is expressed in capital letter and one of the allele is in small letter. And the second genotype for female non-bald is XX homozygous recessive BB. Homozygous recessive is expressed using small letter allele. For the male bald, there are two genotypes, XY homozygous dominant BB, and the second one is XY heterozygous gene pair BB. And for the male non-bald, we have the XY homozygous recessive BB. Note that with heterozygous gene pair for baldness, which is expressed in capital letter B and small letter b, males express baldness while the females do not. Okay, remember, in a sex-influence trait, it is where there is variation on how the males and females express a gene or a trait. Okay, let's proceed to sex-limited trait. So what are sex-limited traits? Sex-limited traits are expressed exclusively in one gender. And these traits may be located either in the sex chromosome or the body chromosome, also called the autosomes. And third, these traits control the expression of the primary and secondary sex characters. When we say primary sex characters, these are present at birth. Example, for the male, the testes; for the female, the ovary. While the secondary sex characters are present during puberty or sexual maturity. For example, for the female, we have the development of mammary gland, menstruation activity, widening of hips, and for the male, so we have the deepening of the voice, development of the Adam's apple, and muscular development. What are examples of sex-limited traits? Example of these traits which are exclusive only to female, so we have the milk production or lactation, the egg production, and giving birth. Some animals also exhibit sex-limited trait. For example, for the male goats and male sheep, they exhibit a horn, and for the peacock, there is a feathering trait in which during courtship, the peacock extends its long, colorful tail to attract the female partner or the peahen. Cock feathering trait is also a sex-limited trait. So, the male birds and the female birds have different or exclusive feathering traits. In milk production or lactation, though the males have breasts, but they don't have the ability to produce milk because they don't have the hormones that enable the gene of lactation phenomenon to turn on. That is why milk production is present only among the female mammals. This is all for today, students, for our science lesson. Thank you very much for your time for this RBI segment. Thank you, DepEd Dapitan City, headed by our Superintendent, Dr. Felix Romy Triambolo. And thank you, LGU Dapitan City, headed by the City Mayor, Honorable Rosalina Garcia Haloshos. Grade 9 students, if you want to learn more about our lesson, you can follow my YouTube channel, Shermay Fernandez, for more educational videos, the Non-Mendelian Genetics. Rise and shine, Ido Calidad. Stay safe, everyone. Bye-bye.